In this video, we're going to focus on how action potentials spread down an axon, and particularly how myelin affects that process. So if you haven't watched it yet, make sure that you go through the previous two videos in the playlist that are before this, where we talk about setting up resting membrane potential in a neuron and generating an action potential to begin with. And so in the last section, we left off at the idea that we are starting an action potential, specifically at the axon hillock, which really just means the starting point of an axon where it branches off the cell body, right? So as a quick recap, we got excitatory or inhibitory postsynaptic potentials coming into this cell body. And all of those signals get integrated at the axon hillock. That's where you really have to decide whether you hit threshold or not. And remember, threshold is this idea that if we cross this line at around negative 55 millivolts, that's the amount of electricity that it takes to open up those voltage-gated ion channels that you find all throughout axons. And if you open those voltage-gated ion channels that we saw back here, you expect that signal to spread all the way down the axon. And the way that it spreads is like a wave, because each of these channels creates a chain reaction that opens the next channel. And when that one opens, it opens the next channel, right? Because the thing that opens the channel is voltage. And when the channel opens, what do you get? Even more voltage, because sodium flows into the cell, right? And so we have this wave going all the way down the axon. But the clarification is, where exactly are those channels and how does myelin come into play? So as an intro, we need to think about what actually makes an action potential travel faster or slower down an axon. Okay, so the first thing is, don't mix up velocity or speed of an action potential with frequency, right? Speed or velocity is asking how fast in like miles per hour or meters per second is the action potential moving down the axon. That is not the same thing as frequency. Frequency is asking how many action potentials per second does that neuron send, right? So this is how many are being sent. This, or velocity, same thing, are how fast they're traveling, right? And we've got a few different considerations. In the physics of electricity, how fast electrical current flows is a matter of something called resistance. And resistance is exactly what it sounds like. Anything that prevents electricity from flowing. The same thing's true for the flow of liquids. And so, like drinking water through a drinking straw, it's the same idea. You have to care about the diameter or the thickness and also the length. So in this equation, R is resistance, L is length, A is area, which means like cross-sectional surface area. But in principle, you can think of this being the same thing as thickness because they're directly related to each other. If the axon gets wider in diameter, its cross-sectional surface area is also going to go up. And this is just a constant that we don't really have to worry about. And so the takeaway from this is, if your goal is to have your resistance be low, then that means the fastest traveling axons are going to be ones with a short length, because we would want this top number in our fraction to be low, and ones with a big cross-sectional area and be thick, because we would want this bottom number to be high. Because in the end, if you divide a small number by a large number, you get a low resistance. The problem with this is you can't really control the length of your axon because it is what it is. The axon has to go from your brain to your spinal cord, and from your spinal cord out to arms and hands and eyes and feet and any structure that's in the peripheral nervous system. And area, yes, we could make the axon thicker, but then it's just going to take up more space, right? And it doesn't really make sense to have a bundle of axons that's incredibly thick, because it's just going to make your entire body larger. Okay, so how do we get around this? One way that you can make action potentials travel faster without having to make the axons shorter or thicker is by adding myelin. And myelin is really just a lipid membrane-like substance that's wrapped around axons, and it acts like an insulator. And so during neural development, this is our axon. This is the nucleus of the cell that is going to become the myelin sheath. So in the central nervous system, we call them oligodendrocytes, and in the peripheral system, we call them Schwann cells. but it's the same idea. These cells are going to latch onto the axon and start crawling around it, almost like the shape of a snail shell. And every time the cell wraps around one time, it's leaving a layer of its membrane deposited around the axon. And since cell membranes are made mostly out of lipids, that's what myelin is composed of. This is the reason that white matter is white in the brain. All the myelin is made of fat, and fat or adipose tissue is white. Okay, so when you look at this picture from an electron micrograph, notice all of these lines, right? This is the middle of the axon right here. If the axon was like a tube, we're staring down the tube, and all of these lines that you see like tree rings, each one of those is one layer of membrane that these cells left deposited around the axon when it developed. And for right now, the quick takeaway is adding this myelin makes the signal, the action potential down an axon, travel faster. The reason for that is a little bit weird. So we say that this type of signaling, when myelin is present, is called saltatory conduction, right? And the reason for that is that on an axon, right? So in this picture, right, this is our cell body back here, right? This would be the nucleus of the cell. These are dendrites over here. And so this is our axon hillock. This line is our axon. And what they're trying to show you is myelin is not continuous. Each one of these Schwann cells, if we were in the PNS, constitutes one segment of myelin. So this area would be myelinated, and this cell also myelinated, but you see we leave tiny gaps in between. Those gaps are called nodes, specifically nodes of Ranvier, just after the guy that discovered them. And the key to this, if we zoom it in up here, is to see that wherever you find myelin, in this picture, myelin is represented by the yellow structures surrounding the axon. There are no channels there. And since it's an insulator, that's the entire point. We don't want ions moving back and forth across this membrane. We want them to stay trapped in the axon. All right, so all of those voltage-gated channels that we talked about in the last video that are in an axon, you only actually find those ion channels in the nodes in between. And so what you expect to happen is at the axon hillock, we hit threshold, voltage-gated sodium channels open, and sodium diffuses into this axon. That sodium will diffuse across this myelin segment through the axon and cause the sodium channels in the next node to open because they hit threshold, right? And then they let sodium in, and you get diffusion to the next one. And this is going to keep jumping from node to node to node all the way down the axon, right? This is called saltatory conduction because that word saltatory in Latin is connected to the word to jump or hop. And so it's not a continuous flow down the axon, it's a hopping from node to node. And the more myelin there is, the faster the signal travels. All right, the reason for this is kind of counterintuitive. If you remember back in this equation, we said that if you can make the axon shorter, it'll go faster because you're decreasing your length. What those myelin nodes are doing is basically taking one really big long axon, right? Axons can be several feet long, and it's breaking them up into smaller and smaller sections. That way you get to plug the small length number for each little segment into this equation, as opposed to the enormous number for the entire axon. I know it doesn't make sense that should make action potentials go faster, but it does. And that's easy to see in patients that have demyelination diseases like multiple sclerosis. They lose their myelin, their action potentials travel too slowly, and it really disrupts their coordination and their ability to contract muscles throughout the body because now the signals coming from their brain aren't getting to the muscle as fast as the brain expects them to, right? And they have weakness and discoordination. All right. One other thing to connect this idea to is the different components of the nervous system. In a previous video, we went through this flowchart and talked about different parts of the nervous system. And so think about where would you expect to find the most heavily myelinated neurons, right? Places where speed matters. So typically, somatic neurons, remember somatic sensory means touch and pain and temperature from skin. And somatic motor means that we're contracting skeletal muscles. That's where speed matters, right? This is reflexes, right? You touch a hot stove and pull your hand back. You step on something sharp when you're walking barefoot and have to retract your foot so that you don't get a nail through your foot. Those are things where fractions of a second matter, right? Or the visual pathway. If a baseball is flying at your head and you have to dodge it, tenths of a second make a big difference as far as safety goes. In the visceral sensory side or the autonomic motor, speed isn't as big of a deal. We're talking about turning your stomach on to digest a meal, changing your heart rate, adjusting breathing rate. If those things happen in 1 second as opposed to a fourth of a second, it's just not a big deal. And so you tend to see the heaviest myelination in somatic portions of the nervous system, not so much in the autonomic or visceral portions. All right, so as you're going through and studying this stuff, try to put it in context. Remember, how does myelin affect action potentials? Where do you find those ion channels? And I find it very helpful to draw pictures as you do this. And so as you're studying, don't just stare at the slides over and over again. Draw out a neuron, draw myelin segments, draw in these ion channels, and explain to yourself, act like you're teaching it to someone else. How does this entire process work from start to finish? All right. Okay, hope this was helpful. Again, the videos will make much more sense if you watch them in order. So follow the playlist, subscribe to the videos. That way that you get all the content and they're not just isolated by themselves. After this, the next video in line is going to move on to the details of what's happening at synapses and the release of neurotransmitters.